Greetings everyone, I'm glad to meet you again today. As I know you remember, I'm Judith Musevi, taking you through the biology. Our topic was the circulatory system, where remember we had said it has got three components. We said circulatory system comprises of the blood, it has got the heart, and it has got the blood vessels. For today, we shall look at the blood. Now the blood, you said it is basically a fluid connective tissue whose main business is to transport materials in the body. It is made up of two main parts. That is, it is a solid part and a liquid part. You have got the fluid part of the blood, which is basically the plasma. It makes up 55% of the blood. And then you've got the solid part of the blood, which is the blood cells, which makes up 45% of the blood. Now to look at this liquid part of the blood, that is the plasma. So the plasma is a pale yellow fluid. It contains all the things we expect to find in the blood in their dissolved form. So we find the plasma being composed of water, which is the main component because it is a fluid. It has got these mineral salts in their dissolved form. So they tend to exist in the plasma in the form of the ions, the component ions. It contains the food materials. The food materials will be present in the blood in their simplest form, that is after digestion is complete. So we expect to find the carbohydrates present in the form of glucose. We expect to find the proteins in their simplest form in the form of amino acids. And we expect to find the fats in the form of fatty acids. We also expect the blood to contain the plasma proteins. These are proteins that are found in the blood fluid part, that is the plasma, in very large amounts. The reason why they are called plasma proteins. And there are three main types of them. We have got the albumins, we have got the globulins, and you have got fibrinogen. We shall talk more about them in just a little while. We also expect to find in the blood the respiratory gases. The respiratory gases here specifically, we are talking about oxygen and carbon dioxide. We also expect to find organic waste materials in the blood, where the main waste material we find there is urea, which is a waste material from the metabolic process of the breakdown of the proteins. We also expect to find hormones in the blood and many other things such as vitamins, such as antibodies, and so on. Now for the plasma, basically it has got a pH of about 7.5 in most places. Now, to go back to the plasma proteins that we talked about, that is the albumins, the globulins, and fibrinogen. They are all synthesized in the liver, which is a vital organ, and these plasma proteins tend to vary in terms of size and also quantities that they are found within the plasma. The albumins, to start with, they are the smallest in size. They are present in the blood in the largest amount. Because of the large quantities, their main role in the plasma or in the blood as it is that they help to maintain the osmotic pressure of the blood, and they also act as protein carriers for basically steroid hormones. And then we have the globulins. Globulins are larger in size than the albumins. They are present in the plasma in smaller amounts compared to the albumins. They are also expected to be synthesized in the liver, except a specific type of globulin that we call the gamma globulin. When it comes to the role of the globulins, gamma globulins specifically act as antibodies, and they are produced in the lymphoid tissue. The other globulins, other than the gamma globulins, they act as carriers for hormones, specifically peptide hormones, and also mineral salts. Now for the fibrinogen, these are the largest in size compared to the others, and they're present in the blood in very small quantities. For them, while the albumins are helping with maintaining osmotic pressure of the blood and acting as carriers, and you've got the globulins acting as carriers and also carrying around mineral salts, and antibodies as well. For fibrinogen, they play a role in the clotting of the blood. For them, they are important in blood clotting, where they tend to form what we call the fibrin threads in the process of clot formation. Now, for the case of the plasma, that's the much we're going to say about them for today. And we now move on to the blood cells. Now, the blood cells, there are three main types of blood cells. That is the red blood cells that are also called erythrocytes. We have got the white blood cells that are also called leukocytes. And we've got the platelets that are also called the thrombocytes. These are the main types of blood cells that we have. Any other blood cell that we have is basically a modification from any of these three that we have. Now, to start with the erythrocytes, or the red blood cells, one thing to note is that all the blood cells are generally manufactured or produced in the red bone marrow. So for the red blood cells, like every other blood cell, they are manufactured in the red bone marrow. The process by which they are manufactured is called erythropoiesis, and this process is controlled by a hormone that we call erythropoietin. For the red blood cells, in terms of structure, they tend to be indented on both sides. A curve on the inside, we refer to that as a concave. So because they are curved on both sides inwards, we now refer to them as biconcave discs. In their young stages, the red blood cells have got a nucleus, but when they mature, they do not have any nucleus at all. Now, for them to have their biconcave shape with even lack of a nucleus, they need vitamin B12, which is necessary for their maturation. Vitamin B12 enables the red blood cells to know what size to grow to and even what shape to take. So it helps them to get the right size and the right shape, which is important in terms of their function. For the red blood cells, they contain a red oxygen-carrying pigment that we call hemoglobin. Now, hemoglobin consists of a protein that we call a globin protein and what we call a heme group. Now, what we call a heme group basically is a molecule that contains iron in it. It generally contains iron in the oxidation state of iron 2. We all know iron is an element with many oxidation states, but the one that we find in the heme group is iron 2. Each hemoglobin molecule has got the globin protein and 4 of the heme groups we are talking about. The heme groups provide the sites where oxygen attaches to when it attaches on hemoglobin. So we say, therefore, the heme groups are the active sites for the attachment of oxygen gas. Now, the iron in the heme group, it loosely attaches or forms a link with the oxygen molecules. A weak link such that the oxygen molecule can attach on the iron without oxidizing the iron. And then the heme group will carry the oxygen on the hemoglobin molecule to the body tissues in this combined form in what we refer to as oxyhemoglobin. The red blood cells will they do the business of carrying oxygen to the body tissues until it reaches a point where they can no longer carry the oxygen to the body tissues. When this happens, we say the red blood cell is worn out. When a red blood cell can no longer carry oxygen, we say it is worn out. So and when it is worn out, it is destroyed in the liver and in the spleen by some cells there we call the macrophages, and the red blood cells are destroyed by the process of phagocytosis. So the red blood cells have got a period of being formed that we call erythropoiesis, and there is a time when they are worn out and they are destroyed by phagocytosis. So they have got a period by which they will exist as they carry out their function and activities. So that period of time from the formation of the red blood cell to its destruction because it is worn out gives us the lifespan of the red blood cell. So for the red blood cells, we say they've got a lifespan of 100 and 120 days, after which they are destroyed in the liver and spleen by phagocytosis. as the red blood cells that are worn out are being destroyed, they have this red pigment that we call hemoglobin. So you expect hemoglobin also to be broken down, and as it is broken down into two waste pigments that we call bilirubin and biliverdin. The two waste pigments are normally put in the bile juice in the gallbladder so that they can be excreted through the body's excretory system. So because they are normally put in the bile in the gallbladder, we refer to the two pigments as the bile pigments. So once the pigment is broken down and the red blood cell is broken down, the waste material is carried away by the blood. But although the red blood cells are formed by erythropoiesis, destroyed when they are worn down or worn out by phagocytosis, we find that the number of red blood cells that an individual has is more or less constant. Now, why this number is constant is because the rate of production of the red blood cells is equal to the rate of destruction. In other words, the rate of erythropoiesis is equal to the rate of phagocytosis of the red blood cells. The role of the red blood cells basically is transportation of the respiratory gases. And we tend to find, because the males and the females have got a different body structure, we have got a difference in the number of red blood cells found in the male and that found in the female. So that generally, the males have got a higher number of red blood cells compared to the female. Reason being that the males have got more of the muscle tissue, which requires energy because it is active. So they need more red blood cells to carry oxygen to this active tissue that is the muscle. The female, on the other hand, have got more of fatty tissue, which is a storage tissue. It doesn't require much oxygen, so they have got fewer red blood cells. Now, having looked at the structure of the red blood cells, describing this by concave discs, this is what the red blood cell looks like. You can see it is indented on the inside. That is what we refer to as the concave curve, and it is on both sides. The reason why we call them biconcave discs. All red blood cells are similar, so you don't have different types of red blood cells. Their function, as we said, is basically transportation of the respiratory gases, which is oxygen and carbon dioxide. To look at a brief look of how the red blood cells transport the respiratory gases, we start off with the transportation of oxygen. Oxygen diffuses into the red blood cells across the thin cell membrane, and then it combines with the hemoglobin on the heme groups that we talked about, forming a compound we call oxyhemoglobin. Each of the four heme groups combines with an oxygen molecule in a loose link without oxidizing the ion in the heme group. So we find that the combination between oxygen and the heme groups to form oxyhemoglobin happens in the lungs, where the air comes in with the oxygen for this gaseous exchange to take place. Now we are expecting that the oxygen will be carried to the body tissues by the red blood cell in this combined form as oxyhemoglobin. In the body tissues, we expect the oxyhemoglobin to release its oxygen to go to the body tissues. So now we can represent that in a simple equation where we have a hemoglobin molecule. Because it has got four heme groups, it can carry four oxygen molecules at a go to give us the combined product, oxyhemoglobin, which is the means by which oxygen is carried to the body tissues. And we expect that to happen in the lungs. In the body tissues, we now expect the oxyhemoglobin to dissociate to give us back the free hemoglobin and the oxygen, where the oxygen now goes to the body tissues. So we expect the reverse to take place in the body tissues. The reaction takes place quite easily because the link between oxygen and hemoglobin is a loose link, just like we said. Now, hemoglobin has got a high affinity for oxygen, which is normally shown by the oxygen dissociation curve. And the reason for this is because when one oxygen molecule attaches to one heme group, it causes the protein molecules in the hemoglobin to alter or change their positions, making it easier for the rest of the oxygen molecules to attach. So for that reason, we say hemoglobin has got a high affinity for oxygen. To demonstrate the high affinity for oxygen, we use the oxygen dissociation curve, and this is what the oxygen dissociation curve looks like. The steep part here tells us how quickly hemoglobin can pick up the oxygen. And it also shows what would happen in case you have got less amount of oxygen and you've got more of carbon dioxide, which is the other respiratory gas. Too much carbon dioxide will cause the curve to shift basically to the right, and that means the hemoglobin will take up less oxygen. Less of carbon dioxide, the curve will end up shifting to the left, and it means the hemoglobin will take up more of the oxygen. So that curve quickly demonstrates to us the affinity or high affinity of hemoglobin for oxygen gas. Now, for the transportation of carbon dioxide, hemoglobin that is found on the red blood cells doesn't have a high affinity for carbon dioxide. So for the red blood cells to carry carbon dioxide, it requires assistance, and that is brought in by an enzyme that we call carbonic anhydrase. So you have carbon dioxide from the body tissues. Because you are expecting this to take place in the body tissues, will diffuse into the red blood cells with the help of the carbonic anhydrase. And it will combine with the water to form carbonic acid, as indicated in this equation. Carbon dioxide combines with the water to form carbonic acid. Now, the carbonic acid is a weak acid, but it will undergo dissociation to give us hydrogen ions and bicarbonate ions. The bicarbonate ions will diffuse out of the red blood cell and into the plasma, carrying the carbon dioxide with it. So for that reason, we find most of the carbon dioxide, therefore, is transported in the form of bicarbonate ions. As the bicarbonate ions are carried away, the hydrogen ions are left behind, and they can cause the red blood cell to become acidic in nature. When the red blood cell becomes acidic, that can lead to the death of the red blood cell. So we need the hydrogen ions formed from the carbonic acid to be taken care of, to avoid the cell becoming too acidic and dying. So now we have the hydrogen ions are buffered by hemoglobin. And we find, because the hemoglobin goes to the body tissues as oxyhemoglobin, the high acidity in the cell actually causes the oxyhemoglobin to dissociate very fast to free hemoglobin and release its oxygen, which goes to the body tissues. The hemoglobin then becomes free to go and buffer the hydrogen ions. So you end up with the hemoglobin now combining with the hydrogen ions to form very weak hemoglobinic acid. And this prevents the red blood cell from becoming too acidic, therefore preventing the death of the red blood cell. Now, that buffering done by hemoglobin can also be done by the plasma proteins. When the plasma proteins do the buffering, they end up forming proteinic acid, which is also a weak acid. So we find for carbon dioxide, it is transported in a number of forms. It can be transported in the form of bicarbonate ions, which is the form by which most of the carbon dioxide is transported in the blood. It can also be transported while combined with hemoglobin, although a small amount, and the product they'll form here is carbaminohemoglobin. It can also be transported while combined with the plasma proteins, where the product here they'll form is carbaminoprotein. And you have a small amount which is transported while simply just dissolved in plasma. And for the transportation of the gases, that is the far we shall go, and for the red blood cells. Now on to the white blood cells or the leukocytes. Just like the red blood cells, the white blood cells are also formed in the red bone marrow. They are formed by the process we call a leukopoiesis, also controlled by a hormone that we call leukopoietin. For the white blood cells, we have got different types of white blood cells. And all of them have got a nucleus when they are mature. So they've got a nucleus in maturity. Now for the white blood cells, we have those that have got granules in the cytoplasm, and have got those without granules in the cytoplasm. So we divide the white blood cells into two main categories. Those with granules in the cytoplasm, that we now call granulocytes, and those without granules in the cytoplasm, that are now called agranulocytes. Agranulocytes meaning absence of granules. For the white blood cells, therefore, unlike the red, we have got different types of white blood cells. To start off with the agranulocytes, the white blood cells that don't have granules in the cytoplasm. For these ones, agranulocytes do not have they have a nucleus that doesn't have any lobes. They don't have granules in the cytoplasm. For the agranulocytes, we have got two types of agranulocytes, the lymphocytes and the monocytes. To differentiate the two, the lymphocytes tend to be smaller in size. They have got a spherical nucleus that is large in size and almost covers the entire cell. For the monocytes, they have got a bean-shaped nucleus and they have got vacuoles in the cytoplasm. Now for the lymphocytes, something to note about the lymphocytes, they are formed in the red bone marrow like every other white blood cell, but they can also be formed in the lymphoid tissue, such as the spleen, the lymph nodes, and so on. For the other category of white blood cells, that is the granulocytes. For the granulocytes, these are white blood cells that have a nucleus with the lobes in it. So we say they have a lobed nucleus and they have granules in the cytoplasm. There are three types of granulocytes: the neutrophils, the eosinophils, and the basophils. To differentiate the three, the neutrophils have got 3 or more lobes in the nucleus, and then they have very fine granules in the cytoplasm. The eosinophils, on the other hand, they have 2 lobes in the nucleus, so we say the nucleus is spectacle-shaped, and they've got larger granules in the cytoplasm. The basophils, they've got a kidney-shaped nucleus and very large granules in the cytoplasm that almost obscure the nucleus. So for the white blood cells generally, looking at both granulocytes and agranulocytes, we end up with 5 different types of white blood cells: the lymphocytes, the monocytes, the neutrophils, the eosinophils, and the basophils. Although the white blood cells are different, they all play the same role of body defense. So to differentiate our white blood cells, we have got, this is what the lymphocyte looks like. No granules in the cytoplasm, a large spherical nucleus. This is the monocyte, which has got a bean-shaped nucleus. That is the neutrophil with 3 or more lobes in the nucleus. This one has got 3. This is the eosinophil with 2 lobes in the nucleus. And this is the basophil with a bean-shaped nucleus. So just like we said, all of them, they may be different, but they do the same role of body defense. But because they are different, they defend the body in different ways. The lymphocytes will defend the body by producing antibodies. So very important in protecting the body against viruses or other disease-causing agent that is an antigen in nature. The monocytes and neutrophils, they are phagocytes, and they take in solid material and basically destroy them by phagocytosis. So for the neutrophils and monocytes, they defend the body by phagocytosis, making them very useful in defense against bacterial infections. Eosinophils, on the other hand, they defend the body against parasitic infections, and they also play a role in regulating allergic reactions. They can generate an allergic reaction where none is showing, and they can also reduce an allergic reaction that is an over-response that can end up causing secondary infections. So they'll basically generate allergic reactions by producing a substance we call histamines, and they can reduce an allergic reaction that is too much by producing substances we call antihistamines. By doing that, they balance the allergic reactions. Basophils, on the other hand, not very clear how they defend the body, but they tend to produce histamine, which is an anticoagulant, rather histamine, which is an inflammatory response, and also heparin, which is an anticoagulant at the site of injury. So because of the way they defend the body, the white blood cells can be used for diagnostic purposes, where if there's an increase in the number of white blood cells, a specific one, then that can give us an indication of the type of an infection an individual has. Too many of the lymphocytes generally could indicate a viral infection. Too much of monocytes and neutrophils, especially so the neutrophils, is a likely indication of a bacterial infection. An abnormally high number of eosinophils indicates either a parasitic infection or an allergic reaction. And then we have the platelets, the third type of white blood cell. Platelets are also formed in the red bone marrow by the process of thrombopoiesis, controlled by a humoral substance that we call thrombopoietin. They are formed by the breaking down of large cells found in the bone marrow that we call the megakaryocytes. Because they are formed by fragmentation of a large cell, we find that platelets, therefore, they are very small in size, they are irregular in shape, don't have a specific shape, don't have a nucleus, and they do have a granular cytoplasm. For the function of the platelets, they play a role in preventing excessive loss of blood. And they do that in three ways. One, at the site of injury, many platelets will come together, form a solid mass of platelets, what we now call a platelet plug, therefore preventing excessive loss of blood. They also release a hormone that we call serotonin at the site of injury, which causes vasoconstriction, or the narrowing of blood vessels in the injured area, causing less flow of blood to the injured area, therefore preventing loss of blood. And thirdly, they can cause the formation of a clot, which prevents the loss of blood. To look at briefly how they cause the formation of a clot, we have the damaged tissue at the site of injury and the platelets. They'll produce a substance that we call thromboplastin, also referred to as thrombokinase. Now, the thromboplastin acts on a substance called prothrombin in the presence of calcium ions, converting the prothrombin to become active thrombin. The thrombin then acts on the plasma protein fibrinogen that we talked about and converts it into insoluble fibrin fibers. The fibrin threads that are fibers that are formed then wrap themselves around everything around them. That is the blood cells, the red, the white, and the platelets, and also the plasma, forming a solid mass that we now call the clot. And the clot ends up blocking the blood vessel at the site of injury, preventing further loss of blood. When we talk about clotting of the blood, what is also referred to as blood coagulation, we already talked about the platelets, but there are actually a number of substances in the blood that play a role in clot formation. Some will promote clot formation, we call those ones procoagulants. Some inhibit clot formation, we call them the anticoagulants. Whether a clot is going to form or not depends on the balance between the two, the procoagulants and the procoagulants. When we don't have an injury, activity of procoagulants is higher, so a clot is not formed. When there is an injury, the activity of the procoagulants is basically higher, so a clot is formed. When we don't have an injury, the activity of the anticoagulants is higher, so a clot is not formed. Yeah. Once an injury starts to heal, we no longer need the clot, so it is normally broken down. The process of breaking down a clot, we simply refer to it as fibrinolysis. We have some enzymes produced where the clot is, they digest the clot, and the waste material is carried away by the blood. I'd also like to talk about macrophages. Now, this is a phagocytic cell that actually develops from the monocyte. So for the macrophages, these are like the monocyte, but larger in size. That means they can therefore take in larger particles and digest larger particles. For that reason, macrophages also play a very big role in body defense, and they can also take in and destroy worn-out cells, damaged cells, disease cells, and so on. So therefore, they play a role in body defense. There are a number of organs in the body that have got a high concentration of macrophages, such as the liver, the spleen, and so on. The network of these organs with a high concentration of macrophages gives us what we call the reticuloendothelial system. To summarize the function of the blood now, having said all that we have about it, we give it functions according to where it plays a role. So function number 1, we give it the role of respiration because of transporting the respiratory gases. We give it the role of nutrition because of transporting the food materials. We give it the role of excretion because of transporting the waste material from the body tissue cells to the excretory organs. We give it the role of protection because of having the white blood cells that play the role of protection. We give it the role of regulation because of transporting the hormones which regulate many processes in the body, including growth and development. We give it the role of temperature regulation because it helps in maintaining the body temperature through radiation, either by vasodilation or vasoconstriction. When temperature is high in the body, the blood vessels undergo vasodilation, bringing blood near the surface, and the body loses heat to the environment by radiation. When the body temperature is low, the blood vessels undergo vasoconstriction, so preventing loss of heat from the body by vasoconstriction. And with that, we summarize the lesson for today. Thank you for being with me for this time, and stay safe.
These televised lectures supplement our robust online learning going on our MKU online platform. You can view more on our televised lectures via our online platform. We are in a digital era, and Mount Kenya University knows this. The following are the steps to follow so as to complete your online application. Download the application form from the website www.mku.ac.ke. Attach copies of your academic certificates and ID. Pay the application fees via M-Pesa pay bill number 270988. Your ID is the account number. 2,000 shillings is the charge for a postgraduate. You can also deposit in the bank accounts provided on the website. Then email all the above to apply@mku.ac.ke.